introduction, and thanks to my co-organizers for the opportunity to speak at this meeting. The title of my talk is Cell Therapy for Viral and Non-Viral Epithelial Cancers. This is my conflict of interest slide. So development of cell therapy for epithelial cancers is the primary focus of my lab and my clinical research effort. The short-term goal is to investigate principles of cell therapy in epithelial cancers using HPV-associated tumors as a disease model. The long-term goal, however, is to discover and develop effective cell therapies for HPV-associated cancers and other malignancies. So I just want to take a minute to talk about the importance of elucidating principles. So in vitro studies and animal models point to wide-ranging potential treatment limitations for cellular therapy in solid tumors. And these include trafficking, T-cell persistence, T-cell function, tumor microenvironment. But it's really critical to understand the actual limitations in humans, especially as this is a field that's really in its infancy of cell therapy, and in particular cell therapy for epithelial cancer. So to do this, we need in humans a model that's not limited by on-target toxicity and a model that's not limited by target antigen loss. And really the ideal model for this, I think across all of epithelial cancers, is the targeting of HPV antigens in HPV-associated cancers. And that's because the HPV oncoproteins, it's E6 and E7, are shared between tumors from different patients. They're tumor restricted, in other words, not expressed by healthy tissues, and they're uniformly expressed. And this is a really unique constellation of characteristics in epithelial cancers and a huge advantage for early development and understanding principles. So the first efforts that we had to try to target HPV associated cancers through the HPV oncoproteins were with an approach that is TIL therapy. So for this, we would operate and remove a metastatic tumor. We would grow T cells from tumor fragments, keeping these cultures separate. We would select the T cell cultures that had the best reactivity against the HPV antigens and expand them further and administer them to the patient. Now, the idea here is for a T cell with a T cell receptor that targets an HPV antigen or an epitope of an antigen presented by an HLA molecule to target HPV. What we found, in fact, was that we did give cells that were targeting HPV, but also cells that were targeting other antigens like cancer germline antigens and mutated neoantigens. So this slide shows the clinical results of the first clinical trial of TIL in HPV-associated cancers. So these patients had a mixture of different HPV-associated cancers, including cervical, oropharyngeal, and anal cancers. This is the waterfall plot, and you can see many of these patients had tumor shrinkage, and the patients who had complete responses and partial responses are shown in gray and blue. Two of these patients had their tumors go away completely. That's shown here also in the spider plot. You can see these two patients where their tumors fairly quickly shrank, completely vanished, and remained gone. There was another patient whose tumor was shrinking. He developed a new metastasis that was resected. He went on to have all of his tumor go away also. And these patients are many years out now from a single treatment with TIL with no cancer. I just want to make that point a little bit more. So this is patient 3 with cervical cancer. She was a 36-year-old woman who had received many prior therapies. She had tumors shown at these yellow arrows, and we gave her 1 infusion of TIL, and all of her cancer went away. And with this sort of a cell therapy with a single treatment, she's been able to go about her life. So she's shown here on the left, and this was a year or two after she was treated when her son won the Kansas State Wrestling Championship as a sophomore. He went on to win this as a junior and a senior, and she sent me photos. He's now actually back from college. And my point is that she's been able to go about her life, see her children grow up, and she's doing fine, and this is all with no treatment. This is another patient, patient 6, with a 36-year-old woman with an adenocarcinoma that was HPV-18 positive. She had a very aggressive primary tumor. She had the cancer shown here, the yellow arrows. This tumor here was compressing her ureter. That white dot is a ureteral stent to drain urine from her kidney to her bladder. We gave her a single infusion of TIL, and all of her cancer completely went away, and it remains absent. So this was a photo that her mother took of the two of us in clinic after treatment. And again, she looks happy because she has no cancer, but also because she's receiving no cancer treatment. So we're trying to understand response and resistance. And one important biomarker that we looked at was the reactivity of the cells that we gave against the HPV oncoproteins. And what we found was that there was a correlation between HPV oncoprotein reactivity of the infused cells, that's on the left side here, in responders versus non-responders. And remarkably, really patients who did not show good reactivity against the HPV oncoproteins did not respond to treatment. This was by both a 4-1BB upregulation assay and an interferon gamma release assay. And in peripheral blood, we saw the same thing following treatment. And this is an interferon, LA spot assay. So what did come as a big surprise, however, was examination of what antigens the TIL that we gave were targeting, especially in the patients who had complete responses. So patient 3, again, was the first patient with cervical cancer to have a complete response. And this graph shows her T cell receptors in the infused, or T cell clonotypes in the infused cells. And this is the rank of the clonotypes in the TIL that was given. And the top rank clonotype actually did not target HPV. It targeted a mutated neoantigen, SETDB1. And the same was true of the second clonotype, which actually targeted a different mutated neoantigen. And the third and fourth targeted the same neoantigen as the first. But again, the top 4 are all targeting non-HPV tumor antigens. However, clonotype that was ranked number 6 did target an HPV antigen, and several others also targeted HPV. Patient 6 was similar but different. So similar to patient 3, and again, this was another patient with a complete response, the top ranked clonotype actually did not target an HPV antigen. It targeted KTLC1, which is a cancer germline antigen. And I'll come back to this in a minute as I talk more about bench to bedside and back again research. But the second ranked clonotype did target HPV. So this was a surprise because this trial was intended to target HPV antigens. And it did, indeed, that's what we did. And we had some remarkable responses, but it's not necessarily entirely because of what we intended. There was certainly targeting of other antigens. So here we had responses in 7 of 29 patients. These included cervical, head and neck, and anal cancer. 2 durable complete responses. These are patients really who kind of realized the promise of a cell therapy type approach where we give a single administration of cells and all the cancer goes away. One patient had a partial response that was resected to NED and also is a long-term survivor. There was a correlation between HPV reactivity to the TIL and response, and there was targeting of viral and non-viral antigens. We also are developing TCRs against HPV and KTLC1 that come out of this research, and I'll talk about that more. The FDA has granted breakthrough therapy designation for TIL for cervical cancer, and Iovance Biotherapeutics is developing that with LN-145. But the TIL approach has critical limitations. So, it requires surgery, which actually is a major limitation. It takes a relatively long time to generate a cell product, typically about 5 weeks. And there's no control over the reactivity of the cell product. So if the cells you grow out are not reactive against HPV antigens or other targets that you want to target, there's nothing you can do about it except not treat the patient, which is a very unsatisfying approach. So to develop a more off-the-shelf but still highly personalized approach with more consistent targeting and a more consistent cell product, we've been using a gene engineering strategy similar to CAR T cells. And so for this, from a patient with an HPV-associated cancer, we obtained peripheral blood. The blood has T cells with endogenous T cell receptors. We use a retrovirus to gene engineer these cells to express a T cell receptor that targets HPV-16E7. This is a T cell receptor that we discovered in my lab. We did some work to optimize the expression of this receptor and to develop a retroviral vector for gene transfer. So we use this retroviral vector to gene engineer the cells. They express the E7 targeting TCR. We expand these and give them. And the idea here is for this E7 T cell receptor to target the tumor by recognizing the E7 11 to 19 epitope, which is presented by an HLA-A2 molecule, and E7 is an intracellular protein that has to be processed, and this epitope presented to T cells. This is part of why we use the T cell based approach, is because a chimeric antigen receptor can only engage cell surface molecules, and E7 is an intracellular protein, and this is the natural way that the immune system recognizes these proteins. So the trial design for this was a first-in-human phase 1 3+3 dose escalation design. The treatment was E7 TCR gene-engineered T cells. The patients had to have metastatic HPV-16 cancers and the HLA-A2 allele. Any prior treatment, including checkpoint inhibitors, was permitted. Checkpoint inhibitors are another T cell-based immunotherapy, probably the leading one. And the treatment was with E7 TCR T cells, and you can see here the dose escalation schema. The starting dose is about 5 to 7-fold higher than the therapeutic doses for CAR T cells for B-cell malignancies. And of course, this highest dose is 500 to 700-fold higher. And we did not encounter dose-limiting toxicity, so we were able to escalate to the highest dose cells, and we really just stopped at this dose because it was all the cells that we could reasonably make, and it didn't seem like more cells would be more beneficial. This is the treatment schema for how we treat the patients. We first give them a conditioning regimen of chemotherapy. The purpose of this chemotherapy is to reduce the patient's current immune system to make space for the cells that we infuse. We then infuse the T cells in a single administration, and then we give high-dose IL-2, which is dose tolerance. This slide summarizes the experience in the phase 1 trial. We treated 12 patients. They have the expected different types of HPV-associated cancer, including vulvar, head and neck, cervical, and anal. They typically have multiple sites of disease and multiple tumors at each of these sites. These are patients with very advanced disease. They've generally been treated with multiple systemic agents also, which is consistent with their advanced disease state. And 8 of the 12 had received a prior checkpoint inhibitor. So again, this is a T cell-based therapy. Really, you wouldn't expect patients who are treated with an oncologic drug that acts through a particular mechanism and develop resistance to that to then respond to a second therapy that acts through the same mechanism. So here it's a T cell based mechanism with the checkpoint inhibitors. Dose level is shown here. These cells are very efficiently gene engineered. And we saw responses in half the patients. 6 of the 12 patients responded. And the duration of responses is shown in parentheses in months. But to really understand these responses, you have to look at them. So this is the first patient we treated, who was a 49-year-old woman with vulvar cancer. She'd been treated with 7 prior agents. She had very extensive disease. These are all tumors in her lungs. This is disease in her retroperitoneum and in her right pelvis and her right thigh. This is normal bladder, and these are normal kidney. This is normal ureter. But most of these tumors went away, and this patient ended up having an 8-month response. This is a 59-year-old male with anal cancer. Importantly, he had been previously treated with a PD-1 T cell-based therapy. He had extensive disease. Only some of it fits on the slide. You can see in particular large bulky disease here right above his diaphragm, multiple tumors in his right lung, although he did have tumors in his left lung as well, this big tumor in his kidney. Almost all the disease in his chest completely went away, and this tumor in his kidney shrank considerably. He ended up with a 9-month response. And then finally, this is a 40-year-old woman, again, also had a T cell-based or PD-L1 blockade-based therapy. She had this chest wall tumor that completely went away, this retroperitoneal tumor that completely went away, and this rectal tumor that shrank. We offered her surgery, which she declined, and eventually that progressed. She ended up having an 8-month response. So we're trying to understand response and resistance, and I'll discuss that more in a minute. But it's important to understand the biology of what we saw, and that's illustrated here. This is patient 5. So this was the patient with the extensive disease in his chest, and he had close to 100 discrete tumors. And by the first follow-up at 1 month, the vast majority of these were completely gone. So this is different than checkpoint blockade in that it doesn't take repeated dosing to see tumors eliminated. This acts right away. There is no pseudoprogression. It's also, I also want to illustrate that when these tumors go away, they stay gone. So here we're following the patient out 14 months, And these tumors that went away stay gone. This is patient 12, and it's a similar story. This patient only had 3 tumors, but again, 2 of them were completely eliminated, and they stay eliminated. And again, they're also completely eliminated very quickly. So when we look for response and resistance mechanisms, it's important to understand if the cells that we give persist in the patient's circulation after they're administered. And the color coding here is by dose level, and this is looking at the engineered cells in the blood as a percent of the T cells or as a concentration of the cells. And you can see, especially at the higher doses of cells, there are very high levels of these cells in the circulation. And in fact, they represent the vast majority of cells in some of these patients. And in some cases, we were able to measure this out for close to a year. This is 300 days at the end of the graph. So these cells persist very well in the patients after they're administered. And there's a lot of effort in the field that's gone into engineering the cells to persist better, but that's really, it doesn't appear to be a limitation here. We also looked at whether the cells are reactive against the target antigen. And the way we, in vivo, and the way we did that is we looked at the peripheral blood before treatment, that's the open symbol, and after treatment, that's the closed symbol. And you can see that after treatment, these patients were acquire robust reactivity against the target antigens with a interferon gamma ELISpot assay. So we studied the T cell side of this a lot, and as I pointed out, there are high levels of cells that persist in the patients after they're infused, and these cells are functional. So why do some patients respond and some don't? Well, the answer to that seems to primarily be in the tumors. So these are the patients treated at the top 2 dose levels from whom we were able to obtain biopsies. And patient 4 is the first one shown here. We're looking at the HLA restriction element, antigen processing genes, and interferon response genes. And we're looking at whether there's copy number loss with decreased expression, or damaging mutation. And we found in patient 4, who did not respond to treatment and had a lymph node biopsied after treatment, but it could have been biopsied before treatment because the tumor didn't respond, that there was a mutation in HLA-A201. And again, this is the restriction element. This is the molecule that shows the target peptide to our engineered E7 TCR. So this is like target loss. Patient 5 was even more informative in that the patient had a tumor in the pleural space that was biopsied before treatment. And this tumor completely regressed, and we were not able to biopsy it after treatment because by the first follow-up, it was gone. The patient did eventually recur in the spine, and when the tumor recurred in the spine, it demonstrated HLA-A2 loss. So here we have a control, and that this patient received, both of these tumors were treated with the same treatment, the one treatment that the patient received. Here's a tumor that was responsive and completely eliminated, and here's a tumor that was resistant. And so this seems to be the key difference. Patient 12 was similarly informative. There was a chest wall tumor, was biopsied beforehand. Again, this tumor was gone by the first follow-up and could not be re-biopsied. In the same patient, there was a rectal tumor that shrank. When this tumor recurred, it had a terrifying finding. So this black box is indicating a damaging mutation in beta-2 microglobulin, and the red outline is showing that the remaining copy of beta-2 microglobulin is completely absent. And of course, beta-2 microglobulin is a critical component of the HLA molecule. So just to illustrate that here, patient 5 had a mutation in the alpha-2 domain of the HLA-A2 molecule. Patient 4 had a mutation in the alpha-1 domain of the HLA-A2 molecule. And patient 12 had a mutation in the start codon of the beta-2 microglobulin molecule, and the remaining copy was lost. So these are smoking gun molecular defects that confer resistance to our therapy specifically, but also likely to other immunotherapies, and in particularly loss of beta-2 microglobulin would make this tumor completely invisible to any T-cell based approach. I think this is informative because it clearly shows what the limitations are of this therapy that we're developing, and points very clearly two biomarkers, but I think it also sheds a lot of light on the broader question of why is it that with all the advances that we've had in immunotherapy, that's really the relatively rare patient with epithelial cancer who's cured by T-cell based immunotherapy. So to summarize, there's no dose-limiting toxicity with the E7 TCR T cells. We did not observe cytokine release syndrome or neurotoxicity as major issues, which is very different than CAR T cells. Responses include a regression of extensive disease, a complete regression of many tumors, and we saw responses in PD-1 and PD-L1 refractory tumors. This is 4 of 8 patients. So it's remarkable that this approach you can mediate really fairly dramatic tumor responses after what would be considered the most effective available T-cell based treatments. So we have a new phase 2 trial that's starting at CI&J that uses next generation manufacturing. This will be a 2-week vein to vein time, so patients will be able to start conditioning a week after we perform apheresis, and it will also employ pre-screening for tumor resistance and select only those tumors that do not have mutations that would or copy loss defects that would clearly confer resistance to a T-cell based approach. So I just want to summarize some principles from the study of T-cell therapy targeting HPV oncoproteins in humans. Cell therapy can mediate durable, complete tumor regression. And this is important because we've seen TIL therapy work in metastatic melanoma, but that was really it. Here we can see that the same sort of a T-cell based approach can mediate durable, complete regression of epithelial cancers, including patients with cervical cancer. We also saw that engineered TCR T cells can traffic widely, persist, and eliminate tumor deposits at diverse anatomic sites. So a lot of effort's been devoted towards solving problems related to trafficking and persistence and the tumor microenvironment. But these T cells are able to eliminate tumors located all over the body. The primary limitation appears to be resistance due to tumor intrinsic gene mutations and copy loss events. So this has been observed also in our prior treatments in our prior clinical trial with the E6 TCR T cells in HPV associated cancers that we published in Journal of Clinical Oncology. It's also been observed with mutated P53 TCR T cells in breast cancer, mutated RAS, TIL in colon cancer, and of course, widely reported with standard immune checkpoint blockade therapies. So I think that while there are potentially many limitations to T-cell based therapies in different settings, certainly in this setting of adoptive T-cell transfer, the primary limitation, I believe, is in the tumor and these molecular mechanisms of resistance. So I just want to extend the talk briefly to a new direction and call out some bench to bedside research that I think is interesting. So I pointed out before that patient 6 who received TIL therapy has an ongoing, at this point, 10-year complete response, and that the TIL that we infused had a clonotype that was very dominant, and the top rank clonotype here was targeting this antigen KKLC1, and this clone has a HLA-A101 restricted T-cell receptor. So this KKLC1 antigen is not something that was on our radar prior to this finding, and in fact, it took some really fantastic work by my postdoc, Sonia Stevanovic, to identify the target of this dominant T-cell receptor. KKLC1 is a cancer germline antigen. Its expression in adults is restricted to germ cells, but it's also expressed by cancer. And by PCR, it's been reported to be commonly expressed by gastric, non-small cell lung cancer, triple negative breast cancer, and cervical cancer. By C, these numbers are highly optimistic, but there's still a substantial fraction in our small sample set that we've looked at that have high expression. For example, here's a gastric cancer with 90% expression and a non-small cell lung cancer with 95% expression. So, in some cancers, this seems to be a very uniform, very attractive target. And this restriction element is present in about 25% of the population. So we captured the exact TCR that appears to have been dominant in this patient's ongoing complete response and apparent cure. And we can now engineer anyone's T cells to express this T cell receptor and target cancers that express this KKLC1 antigen. So this is a NSG model testing this T cell receptor. The tumor here is 4156 cervical cancer cell line we generated in my lab. And here we've established it on NSG mice, and we treat them with human T cells transduced with the KKLC1 TCR. And there's a dose response in tumor regression, and highest dose, all of the mice have all of their tumors eliminated. So this is part of the preclinical data that's led us now to a clinical trial at Rutgers Cancer Institute. So to summarize, this TCR was dominant until it cured a patient with cervical cancer. And I think we can say she was cured at 10 years after a single treatment with widely metastatic disease going in. KKLC1 TCR is expressed in KKLC1 is expressed in a high tumor fraction of some gastric and lung cancers, and maybe other cancers were seen fairly good expression in cervical cancer and triple negative breast cancer as well. The KKLC1 TCR T cells mediate tumor regression in mice. And we have a phase 1 trial for gastric, lung, breast, and cervical cancer that we started at NIH. I actually need to update the slide because it's actually active now at Rutgers Cancer Institute, and we're recruiting patients for this trial. For time, I'm not going to summarize the whole talk. I'll just point out future directions. We're looking at biomarkers for prospective patient selection. The biomarkers here are conspicuous. We definitely need to look and make sure that there's no mutation in the restriction element and that there's no copy loss involving the restriction element. We're also going to be looking for biallelic defects in critical molecules like TAP and interferon gamma receptor 1 and JAK molecules. We're also working now with a faster manufacturing process with the 2-week vein-to-vein time. We're trying to move this to an earlier line of treatment, so future protocols will be trying to move up, especially the E7 TCR T cells. And in the lab, we're discovering a library of T cell receptors so that we can target tumors in a polyclonal manner, including through HLA class 2 molecules, which opens up a whole nother doorway to being able to recognize and kill the cells, the tumor cells. We're also working with technologies for targeting immune-edited tumors. And we're trying to improve the toxicity with enhancing T cell function so that we can attenuate or eliminate the conditioning in IL-2. And for time, I will stop here. I've pointed out the lead authors on the papers that I've presented. Thank you for your attention.